Modern DNA technology methods have transformed the field of forensic science, the scientific analysis of evidence for crime scenes and other legal proceedings. Forensic is an argument used for legalities in a court of law, so forensic science refers to the scientific theories in a judicial system. It is also commonly referred to as criminalistics. Throughout the years, science became more important in legal settings, and scientific discoveries were commonly used to decide the outcome of legal matters. By the late 1700s, forensic science was being used as a component to determine guilt or innocence in major crime cases. One of forensic science's primary functions is to study DNA, the genetic coding found on all living things. DNA samples can be acquired from any human or animal and then be tested and compared to other samples for similarities. This process is highly respected and a major consideration for cases in which identity of an individual must be found or established. Forensic scientists don't arrest suspects or evaluate crime cases, but rather they analyze the natural evidence and interpret the results by their lab findings. Forensic science has many uses in criminal justice world today, such as determining cause of death, identifying criminals and bodily remains, tracking the electronic transfer of funds, and investigating fraud and identity theft. The most important application to forensics is DNA profiling, a procedure that analyzes DNA samples to determine if they came from the same individual. An overview of an investigation involving DNA profiling has 3 general steps. First, DNA samples are isolated from the crime scene, suspect, victims, or any other kind of evidence. Then, selected markers from each DNA samples are amplified, which means to be copied numerous times, and this would then produce a large sample of DNA fragments. Lastly, the previously amplified DNA markers are compared, which provides data about which samples are from the same individual. In this presentation, we will be going over an overview of the steps in analyzing DNA evidence, types of DNA evidence analysis, including PCR, STR, Y chromosome, and mitochondrial analysis, microbial analysis, DNA phenotyping, how DNA fingerprinting and CODIS work, and contamination avoidance and DNA success rates.
I will be talking about the steps in analyzing DNA evidence, otherwise known as the steps in the DNA sample process. The first step is extraction. DNA is located within the nucleus of cells. The extraction process is responsible for breaking open the nucleus to release the DNA molecules. These DNA fragments are then placed into a solution. It is important to make sure that the DNA is isolated from all other materials. If it is not, this is something that can throw off the process and make it more difficult to proceed. These unwanted materials are called inhibitors and are most commonly found in hemoglobin and indigo dyes from denim. There are 2 methods for extraction that are most common. The first method is a manual phenylchloroform, otherwise known as organic extraction, and through the use of an electronic system called the Maxwell 16. Organic extraction takes a minimum of 3 hours, and running samples on the Maxwell 16 takes a minimum of 2 hours to extract DNA content. The second step is quantitation. One of the most important things that all DNA testing laboratories check before extracting DNA is that the DNA is recovered from a human rather than another source such as bacteria. This is found through quantitation. Quantitation is where the quality and quantity of DNA present in the sample is measured and assessed. This is also where the amount is determined for how much DNA is needed to be successfully sampled. Quantitation is completed through medical kits and then run through a machine known as a sequence detection system. This takes around 30 to 60 minutes to set up and then approximately 2 hours to run. The third step is amplification. DNA amplification is accomplished through the use of PCR, otherwise known as polymerase chain reaction, and millions of copies of a particular DNA sequence are produced. This copying process is completed by precise heating and cooling of the samples and takes about 28 cycles. This typically takes a couple of hours. For the amplification process, a specific kit is used and then run on an instrument known as a gene AMP PCR system. This process takes 15 to 30 minutes to set up and then around 3 hours to run. The last step is capillary electrophoresis. After amplification is completed, it results in a large mixture of amplified DNA molecules, which need to be separated in order to distinguish molecules from one another. DNA molecules are negatively charged, and once an electric current is applied to the sample, the molecules enter a very thin capillary filled with gel-like polymer and migrate towards the positive anode at the other side of the capillary. The PCR products are then separated by size because the smaller molecules have an easier time traveling through the polymer than the larger molecules. This DNA is then collected on a computer connected to the instrument, and a DNA profile is developed. High-tech genetic analyzers are capable of analyzing up to 4 samples at a time and take about 45 minutes to process.
Using DNA to identify people is complicated because nearly all of our DNA is identical. Forensic scientists use DNA profiling in order to identify possible suspects, victims, or other significant people in a crime case. By utilizing short tandem repeat markers, or STR, forensic scientists are able to identify people through DNA profiling. Some of the uses include confirming familial relations, identifying missing persons, or linking people of interest to crime scenes. STRs are short repeated sequences of DNA, and typically in forensics they use Y STRs, which are the STRs found on the Y chromosome. The sequence of units vary greatly, which makes it effective when trying to identify people. Everyone's DNA is unique, and by comparing 2 samples of DNA, forensic scientists are able to distinguish 2 individuals from one another. PCR, or polymerase chain reaction, is a technique that scientists use to replicate a small amount of DNA, which makes it possible to study the DNA in better detail. This is another method of DNA profiling. PCR method is based off of the mitochondrial gene because of their high copy number per cell. There's a lack of recombination, and they are inherited from the individual's mother. This is advantageous because PCR can be done with small samples of DNA from crime scenes, and additional uses include identifying the biological parents of an individual and other instances outside of forensic science.
The male-specific part of the human Y chromosome is widely used in forensic DNA analysis, specifically in cases where the standard autosomal DNA profiling is not informative, meaning there is limited evidence that could be utilized for other analysis methods that were previously discussed. This method is called Y chromosome DNA analysis. So, why is the Y chromosome specific to this forensic evidence analysis? Well, the Y chromosome is inherited from the father and is conceded to all male offspring. Therefore, the chromosome encodes dozens of male-specific genes, including the sex-determining gene. Y chromosome DNA analysis, or YSRTs, are specifically used when there are multiple source samples or individuals involved. This method is used when autosomal SRT profiles are compromised. Some reasons why this method is used is it can help with the elimination of male suspects from involvement in a crime scene. It is also useful in identifying paternal lineage of male perpetrators and also highlighting multiple male contributors to a trace and providing leads for finding unknown male perpetrators. Thus, other cases which it could be used are in sexual assault cases, cases of missing people, disaster victim identification, population inference, and complex kinship analysis. The Y chromosome loci plays a very important role in the use of profiling DNA in forensics. The Y chromosome loci is located specifically in the non-recombining section, or NRY, of the Y chromosome. This means it doesn't participate in homologous recombination. The SRT loci of the Y chromosome are usually referred to as haplotypes. A haplotype is a collection of alleles that are linked together inheritedly, since recombination does not occur on the majority of the Y chromosome, meaning it's a group of genes with the organism that was inherited together from a single parent. Haplotypes composed of Y chromosomal short tandem repeat polymorphosis. Another method that is used in forensic DNA analysis is mitochondrial DNA analysis. So, since there are two kinds of DNA within our cell, one being within the nucleus and the other through the mitochondria, it can be used specifically for this type of DNA analysis. So the nuclear DNA is a long chain, while the mitochondrial is circular, which has 16,569 base pairs organized in 37 genes. They have non-coding regions and have the kind of variation scientists need in order to distinguish individuals. Unlike nuclear DNA, mitochondrial DNA has a high copy number per cell. The DNA is also more resistant to sample degradation. Mitochondrial DNA, or mtDNA, is inherited strictly from the mother. So similarly to what we previously discussed about the Y chromosomal linking to the males in the paternal line, the mitochondrial DNA links females all the way up the maternal line. Therefore, in situations where the individual is not available for direct comparison with a biological sample, in theory, any related individual would be able to provide a reference sample. The first step in the mtDNA analytical process consists of extracting the DNA from the evidence sample. This may be material such as blood, teeth, hair, etc., which then the PCR or polymerase chain reaction is used to make various copies of the hypervariable non-coding region of the DNA molecule. Flanking primers are used to identify and adhere to the ends of the region wanted to PCR amplify. Then, once a good amount of PCR product is amplified and all the relevant information is extracted about the hypervariable region, sequencing reactions can perform. Here, each complementary strand of DNA are labeled with dye and separated according to size by the machine that specifically reads the sequence of the nucleotide bases. Then, these sequences of the hypervariable regions will allow forensic biologists to be able to differentiate the particular sample. Now, at least two forensic analysis assemble the sequence independently and compare it to a known reference sequence. This process is tested several times with the known sample. The sequences from both are compared to see if anything needs to be repeated. Then, lastly, in this analysis is to search the data for any inclusions or matches of the mtDNA database. A report is written to document any findings of the same type of mitochondrial sequencing. According to our sources, currently the available forensic database of human mitochondrial DNA sequences have over 29,000 sequences available to reference.
I'm going to be talking about DNA phenotyping, which is pretty much predicting the appearance and biogeographic ancestry of a human being. So, scientific definition is it's the science of predicting an organism's observable physical or biochemical characteristics by using only genetic information from DNA sequencing or genotyping. It's also known as a molecular photo fitting. It's primarily used to predict a person's physical appearance and or ancestry for forensic purposes. There's two methods to predict human phenotypes from DNA. They're direct and indirect. Direct is using statistical methodologies to infer trait value and mechanically linking genetic variants with the relevant phenotype's variable expression, which is the preferred method. Indirect is a trait value is inferred by using variants that are associated with genetic components of ancestry and combining these with appropriate statistical methodologies. These all are used in the field of the forensic science. There was an investigation, one of the most famous ones, which involved Kendra Aston and her daughter, Malaysia Boykin, which was in Columbia, South Carolina. It was in 2015, and the use of composite image by police, the first to be introduced from a DNA sample, led to the charging of Kenneth Cantazar with a crime in 2017, which shows just how important these could be for the future.
Fingerprint identification is an exceptionally flexible and versatile method of human identification. The fingerprint is a very certain method for identifying a person because all fingerprints are unique. Even identical twins have different fingerprints. A fingerprint is formed when a finger makes contact with a surface. The finger leaves behind traces of sweat and any other substances present on the finger that a suspect may have touched. These substances are deposited in the characteristic pattern of the ridges present on the fingertip of the donor. Most fingerprints are invisible to the naked eye and require a chemical development process in order to visualize them. Newer processes can obtain much more information about the fingerprint owner, what they've touched, what they've eaten, and even what drugs they've taken. A fingerprint left at a crime scene in forensics called the finger mark will contain not only sweat from a suspect, but also traces of any substances a suspect has touched. This can be very useful evidence if the finger marks contain a victim's blood or explosive compounds, as it instantly ties a suspect to those substances. There are two common ways to collect fingerprints: collecting patent prints and Latin prints. Patent prints are collected using a fairly straightforward method, photography. These prints are photographed in high resolution with a forensic measurement scale in the image for reference. Investigators can improve the quality of the images by using low angle or alternate light sources and certain chemicals or dyes during photography, but this is usually not necessary. Collecting Latin prints is done by dusting a smooth or non-porous surface with fingerprint powder. If any prints appear, they are photographed and then lifted from the surface with a clear adhesive tape. The lifting tape is then placed on a Latin lift card to preserve the print. However, fingerprint powders can contaminate the evidence and ruin the opportunity to perform other techniques that could turn up a hidden print or additional information. Therefore, investigators may examine the area with an alternate light source or apply superglue before using powders. In criminal justice cases, computerized systems are used to search various local, state, and national fingerprint databases for potential matches. Many of these systems provide a value indicating how close the match is based on the algorithm used to perform the search. Fingerprint examiners then review the potential matches and make a final determination. Fingerprint examinations may be conducted by forensic scientists, technicians, or police officers. However, the examiners should have the proper training and experience to perform the task. Currently, many agencies require new examiners to have a 4-year degree in science, either in biology, chemistry, or physics. In addition, agencies may require examiners to become certified by the International Association for Identification, or the IAI. The real investigative power of DNA technology can be realized in its application to cases where a suspect has not yet been identified. DNA technology in the FBI's CODIS database can help law enforcement identify perpetrators or link serial crimes. CODIS stands for Combined DNA Index System, which is a computer software program that operates local and state and national DNA databases. DNA was not used by the FBI or police to solve and fight crime until the 1990s. The FBI began the project now known as CODIS, which following congressional approval and funding became fully operational in 1998. CODIS stores DNA profiles from around the country in a series of local, state, and national databases, all linked via computers, enabling crime labs at every level to share and compare DNA profiles electronically. It was developed to enable public forensic laboratories to create searchable databases of authorized DNA profiles. The goals are to link crimes to each other and identify potential suspects by matching DNA profiles from crime scenes with profiles of convicted offenders. Thousands of CODIS matches have linked cases, and many cases have been solved by matching crime scene evidence to convicted offender profiles. Prior to the 1990s, DNA had not been previously used for crime investigations. Because of this, many innocent people, especially black and brown men, were wrongly incarcerated for crimes they did not commit. The police had previously relied on other factors like eyewitnesses and false confessions to charge and convict perpetrators. Since 1989, there have been tens of thousands of cases where prime suspects were identified and pursued until DNA testing, prior to conviction, proved that they were wrongly accused. According to research done in 2018, 130 DNA exonerees were wrongfully convicted for murders. 31% of these cases involved eyewitness misidentifications and 62% involved false confessions. On October 3, 2018, a man named Horace Roberts was released from a California prison after DNA evidence exonerated him in the 1998 killing of his ex-girlfriend and co-worker. He was convicted by a jury even though no reliable evidence was presented in court. DNA testing of stuff present at the crime scene turned up a DNA profile for an unidentified man, but it wasn't enough DNA to be uploaded to CODIS. Roberts' defense successfully petitioned for everything present at the crime scene to be tested, including the victim's clothing. After there was enough evidence to be uploaded to CODIS and DNA connecting a man named Joaquin Leo showed up. Roberts was later released and compensated for $140 for each day he was in jail, which later added up to $1 million. His story is just one of many, as there are still hundreds of men sitting in prisons all over the country after being wrongly incarcerated.
DNA contamination can happen at any time, including before the sample is collected, while it's being collected, during transport, or at the lab. Hot conditions create a higher risk of contamination. Anti-contamination strategies include lighter suits for scientists. Proper medical PPE equipment should be implemented to avoid contamination. This includes face masks, safety glasses, gloves, an oversuit, overshoes, and a second pair of gloves. DNA samples should be handled with care in order to avoid contamination, and torn gloves are not acceptable. DNA profiling success rates differed between cases, ranging from 50.8% to 7.0%. Success rates for samples was 36.3%. Tooth samples produce a 33.7% success rate.